Hello everyone, in this video I'm gonna introduce and present to you guys a type of enzymes which is familiar with us, popular and very useful in our life, and that enzyme is proteolytic enzymes, also known as protease enzymes. For understanding more about protease enzyme, we will go through 6 parts in this lecture: definition, properties, structure, classification, sources, and applications. First, we have to know what is proteolytic enzyme. As I said before, proteolytic enzyme, also known as protease, are hydrolytic enzymes and are included in the group hydrolases. Besides, it is synthesized by organisms and participates in biochemical reaction. It hydrolyzes proteins to low molecular weight and amino acids. We move on to the next slide to discuss protease properties. Protease is created in biological cells, non-toxic, environmental friendly, participate in reaction both in living cells or when separated from living cells. In an open or closed chain, the product of the former is the substrate for the latter. A reaction can be made occur extracellular asymptotic, consuming very little energy, high efficiency, and speed, highly selective. Is controlled by gene and response conditions. Proteases are not only significant for the growth and reproduction of all microorganisms, it also plays an important role for food processing, medicine, and so on. So let's continue with its structure. Protease enzyme is an enzyme that hydrolyzes peptide bonds, the bond between CO and NH, in protein molecules to release amino acids, peptides or tetrapeptides. Here the structure of protease enzyme. So how do the cultural structure affect? The cultural structure of the protein molecules affects the degradation of substrates under the action of proteases. The monomer and dimer forms of the substrate are easier to degrade than the tetramer form. Compared with animal and plant protease, microbial protease have many different characteristics, and it is a very complex system consisting of many enzymes that familiar structure, shape, and so on. In this slide, we're going to discuss about classification of protease enzyme and how is protease enzyme classified. In order to classify protease enzyme, we have to follow many factors. Based on the position of action of protease on peptides in protein molecules, people divide protease into two main groups: exopeptidases and endopeptidases. Exopeptidases, based on the site of action on the polypeptide chain, are classified into two types: aminopeptidase and carboxypeptidase. Aminopeptidase catalyzes the hydrolysis of peptide bonds at the free nitrogen terminus of a polypeptide chain to release an amino acid, a dipeptide or tripeptide. Carboxypeptidase catalyzes the hydrolysis of the peptide bond at the carbon terminus of the polypeptide chain, and it also releases an amino acid or tripeptide. So how about endopeptidases? Endopeptidases, based on the kinetics of the catalytic mechanism, are divided into four smaller groups: serine proteinase, cytosine, aspartate, And the last one, metalloserine protein acid, containing the OH group. Cytosine protein acid, containing the SF group. In the active site, aspartic protein acid. Most aspartic protein acids belong to the pepsin group. The pepsin group includes digestive enzymes such as pepsin and chymosin. Metalloproteinase is a group of protein acids found in bacteria, mouse, as well as higher microorganisms. Furthermore, we can classify protease enzyme based on the amino acid composition and the optimal pH grade. We have three types: acid proteases. Pepsin renin operate in acidic pH 2 to 4 region. Alkaline proteases, trypsin, chymotrypsin operate at alkaline pH 9 to 11. And the last one, neutral proteases, just as papain from papaya, bromelain from pineapple. It works at neutral pH range 7 to 8. In this slide, I want to share with you where we can find protease enzyme or source of enzyme. Fortunately, protease enzyme exists in many sources around us, such as in plants, animal, or microbial. In plant, it's in papain from papaya, bromelain from pineapple. In animal, it exists in pancreatic trypsin, chymotrypsin, pepsin, and chymosin. And microbial is bacteria and fungi, and so on. We move on to the last part of protease enzyme lecture, application. Protease enzymes have a wide range of applications, especially in food processing. Protease enzymes have a lot of benefits for making new type of products. In this slide, I listed some applications of protease enzyme: cheese making, meat tenderization. In meat tenderization, plant proteases are commonly used for this. Microbial proteases are better choices, which provide controlled hydrolysis. So we continue with protease application. Fish salt is a product that also used in protease enzyme. The enzymes used in fish salt that are mainly used include acidic proteases such as papain and fisin. Baking industry, protease also play an important role to make it. Protease reduces mixing time, enhances ductility and smoothing, produces better heat and vitality. Protease enzymes have a very important role in alcohol production because it helps increase the percentage of substitute materials, reduce fermentation time, increase recovery efficiency. Finished beer and wine methods specify about 30% better improve economic efficiency, reduce production costs. So that's all information about protease enzyme I have talked to you guys. If you want to get more information about protease enzyme, you can access this link below that I used it for my presentation. So if you have any questions or misunderstanding about protease enzyme, and you can leave a comment below in this video. I'll try to answer it as fast as possible. So thank you for listening and watching my presentation. So if you like this video, please thumbs up, share and subscribe my YouTube channel for supporting me to become famous YouTuber in the future.